Okay, so it's my pleasure to introduce Dr. Roy Yanong. I'm very excited to have him here with us today. He joined the University of Florida's IFS Tropical Aquaculture Laboratory in 1996 after serving as staff veterinarian at a large aquarium and fish farm wholesaler. Currently, Roy is a professor and extension veterinarian and provides extension, research, and educational programs in the fish health management and aquatic animal health space, including on-site veterinary assistance and disease diagnostic support for aquaculturists throughout the state of Florida. Roy also works closely with state and federal agriculture and natural resource agencies, veterinarians, and other fish health professionals. His work focuses on baseline health parameters, health management and regulatory issues, disease characterization, disease prevention and diagnostics, chemotherapeutics and biologics, and biosecurity for fin and fish aquaculture. And in collaboration with other faculty and staff at the University of Florida, Roy has been involved with aquatic invertebrate, amphibian, reptile health and disease work. Roy is currently the chair of the Aquatics Working Group for the American Veterinary Medical Association's panel on euthanasia. He's a former member and chair of the AVMA's Aquatic Veterinary Medicine Committee and a past member of the AVMA's Animal Agricultural Liaison Committee. He is also a co-founder and former president of the American Association of Fish Veterinarians, a member of the American Fishery Society Fish Health Section, and the U.S. Aquaculture Society and the World Aquaculture Society, and on the steering committee and chair for the Eastern Fish Health Workshop, which I assume he extends an invitation for next year when we're all back in person. And last but not least, Roy hosts the podcast Aquarium Mania and the fishy part of the Pet Life Radio Network. So without further ado, Roy, we welcome you to our keynote presentation here today. Thank you.
Great, thanks a lot, Nick and Matt and Tom and everybody else involved. This is a great venue, and hopefully I will keep you guys awake. So let's see. All right, do you see my screen okay?
Yes, we do.
Yes, okay, great, thanks. All right, so keynote usually means I'm old, so I'm gonna talk about some of the older cases over the years and talk a little bit about how I got into all of this. I appreciate Nick's introduction. And so this is kind of a little bit of a story of you know, my personal / professional life and how I got to where I am and some of the interesting things along the way. Here, and let's see if I can do this on my computer. It doesn't want to do that. There we go. All right, so a little watered down overview here, a little bit of my beginnings. I've got a really short but kind of really neat aquarium industry video that our lab, spearheaded by Craig Watson, put together for the Florida State Fair a year or two ago, which I think would be really interesting for you all. It's got some nice drone shots, some cool cases. Of course, we have to talk about drugs since Matt Griffin's on the call. And then we'll wrap it up and I'll provide some thanks. So that's kind of our plan. So my family's an immigrant family. My parents and my sisters came over in the 60s from the Philippines. And, you know, my family was pretty poor in the Philippines. So it was kind of that sort of mentality and you know, understanding where, you know, basically there are like several potential jobs that you should get if you come to America. And so, you know, a doctor was one of them, you know, medical doctor. That picture on the lower or the middle kind of left, basically a lot of my dad's and my mother, of course, and a bunch of my aunts and uncles were all doctors. So I think I was kind of, it was assumed I would take over. That's not the kid that was given, but the upper picture is me with my dad. And yes, I did get like a doctor bag when I was probably like 3 or 4 years old. So I think they kind of were pushing me that way. Because of all the things we did growing up, you know, especially living in Chicago, lots of trips to Shed Aquarium. We spent time in Florida going to a lot of the attractions down here, including Miami Sea Aquarium and SeaWorld, watched lots of Jacques Cousteau, and had an aquarium. My very first aquarium is a replica of Is It Below, is the little gourami. I had a bowl with a gourami in it because I read that gouramis could breathe air, and I figured that. So I was wrong. I found out a couple weeks later when my gourami pretty much died, and I'm guessing it was probably ammonia. But at the time, you know, as a 7, 8-year-old, I finally realized that the person at the pet store was not trying to actually pull one over on us. We probably really needed a more specific tank with good filtration. So a little bit about my professional background and kind of academic background. When I was kind of pre-med, I actually did go to Yale and majored in molecular biophysics and biochemistry, which back in the 80s, you can imagine was probably way behind, or definitely was way behind where it is now. I took a couple of years off because I wanted to do some more kind of marine research, and worked at Tufts doing soft shell clam research with the unknown kind of leukemia at the time, which now we know is a transmissible tumor. At the time, we weren't sure what the cause was, or what was, whether it was maybe pollution or a virus, but now we know. I did attend University of Pennsylvania, did a lot of dog and cat, of course, and all that. I did Aquavet 1 and 2, did some really great rotations, including SeaWorld Orlando in February, which kind of told me or made me decide I wanted to move to Florida because of the weather, and it was really great. My first job, I will have to thank my mentor, Greg Lubart, who pretty much helped me get the job at 5D Tropical, the fish farm that Nick mentioned earlier. I was there for 4.5 years. And then at a kind of turning point in my career, I was given the opportunity. I applied to the Florida Aquarium, which was just opened, as well as to here at UF, and was offered both jobs. I decided I really didn't want to do aquarium medicine and was much more interested in ag. So I took the UF job, and I've been here since then. That's an old picture of me actually from 5D. So I did want to acknowledge my mentors, and these are a large group of them. There are others, but these are some of the major mentors that helped me through my vet school and then early career, as well as, even later on. So Greg Lubart, some of you know, or many of you may know, he was a big inspiration for me getting into ornamental, the industry down here in Florida, helped me get my first job. Brent Whitaker, Stoskoff, Ruth, who've done a lot of work with down here in Florida as well, and has been, really important for my development and growth. Dr. Don Apt, Paul Bowser, Ed Nogan, Steve Smith. So definitely a big thanks to all these guys. And some others that I'm not mentioning. And I did want to thank Craig Watson as well. He is the director of our lab here. He's been down in UF working with Ruth with the aquaculture industry. And Craig and Ruth really opened up the industry to science and to working with the University of Florida back in the late 80s. So back when I started in 96, we were not quite as developed as you see in that picture now, which is a drone shot of our lab. But it was pretty much me, Craig, and I think 1 or 2 other people at the time. So we've really gone pretty far since then. So a little about the industry. The Florida Tropical Fish Farms Association actually began in 1964, which is really interesting. Found this out a couple of years ago because I was also established in 1964. A picture of me and my mom. And this is a really nice, that video I mentioned, that kind of gives a little bit of a drone shot overview. So a lot of the farms vary, and most of the freshwater pond production or freshwater production is in ponds. That aerial shot showed. The first farm was in the Miami area in the late 20s, but Eureka Springs in Hillsborough County was the first kind of more local farm. And it was Albert Greenberg, who is a Hall of Famer for Florida, and did quite a bit to bring the industry into our county and surrounding areas. As this mentions here, the first neon tetra actually came over to New York on the Hindenburg. And since that time, both in cans, and we've got cans here, old cans that were used in plastic bags. Fish have been transported from Florida to other parts of the country and the world. This is a really nice drone shot through one of our farms here. I believe this is Urban Tropical in Lakeland. And you can see a lot of burial vaults there used for housing fish. This is kind of an overview shot of some of the ponds with covers. We use covers, various types. These are flat covers that he's using in the winter to try to keep the heat inside. Other farms will use more kind of hoop-shaped covers with a little more of a space, a little bit easier to work. Again, our Florida climate and soils and water are really ideal. Water table is pretty high in this area, so the water will kind of rise as the ponds are dug. This is just a harvest scene of the angelfish, one of the iconic fish of the Florida aquarium industry, and actually on their logo as well. So these folks are using a seine net to capture up the fish. You can see these ponds are significantly smaller than the catfish industry ponds, so much easier to clean and work. He's basically putting the fish into these containers, which will be brought into this holding area where they're going to be held for a certain period of time prior to being sold. So he's just kind of releasing them, a little bit of an acclimation, sort of depending on the facility. Different types of varieties of angelfish. These are kind of his koi angels. And then they're going to be sent to distributors. Some farms are distributing them to larger facilities. So this person here is counting out fish in small numbers. Some farms sell to larger wholesalers. Some facilities, wholesalers are going to send to smaller pet stores in smaller quantities, and that's kind of what this shipment is about. So this facility is probably selling to a pet store that may have smaller quantities required versus a wholesaler. Lots of boxes, lots of fish leaving Tampa, Miami, and other primarily Tampa and Miami. And again, hundreds of species and varieties leaving the state pretty much almost on a daily basis. We do, and I'll touch briefly on marines. These are a lot of the freshwater fish that are in the hobby down here, and I mean in the industry. Quite a few variants of many of these species as well, which have kind of made the hobby much more richer for the hobbyists. And again, thanks to our UF-IFAS Communications for that really kind of cool video. So as I mentioned, we do a lot of marine fish as well, and I've kind of put some pictures up here. The clownfish are doing things like mandarin gobies, some of the dotty backs, et cetera. So there is a larger increasing number of marine fish that are being produced in Florida, in addition to, of course, the freshwater glowfish, which are kind of the first transgenic pet of any type. And quick question: anybody know what kind of fish this is? This is probably not. This is actually a 17-day-old Pacific blue tang in our lab here, and the researchers here, including Matt Griffin, shown in this photo here with Kevin Barden. And Kevin Barden were involved with the first spawn and grow out of the Pacific blue tang, Dory, which was really interesting. It came out about the same time as Dory, the movie, so it was a really great time. And they're still working on trying to make that sort of a cookbook kind of production method for the producers at this time. A lot of this, our marine work is funded through Rising Tide Conservation, Judy St. Ledger and her group, and we really appreciate all that. A little bit about some cases now that we've had over the years. So this is, again, a really old picture of me at 5D Tropical. And as many of you students probably starting to enter the workforce, whatever area you're in, taking the training wheels off is always a little scary. I had some really stupid mistakes made, which I guess are going to be, going to happen, but you try to make fewer and fewer. When I was first hired in '92 by 5D, the owner said, "Roy, you have to solve this issue." And I will give you a heads up, I didn't actually solve it, but luckily I was able to kind of help at least the farm manage it after it was determined to be, after the etiology was determined. So this was in the early 90s, essentially. A lot of the gouramis were having really heavy mortalities, and a few there was at least one farm I know of that actually went out of business. They specialize in gouramis and were having significant 90 to 100% mortalities in ponds. I have 50-80 there, but this guy was losing even more. Typically in the summer and fall months when the fish were ready for harvest, I've got a couple species of gourami up there, and then actually that's like the only picture I have from back then on the upper left, which is sick blue gouramis or opaline gouramis. From the disease outbreak of those years. They were, again, multiple species of gouramis, typically non-remarkable signs. They would darken, hang, have ascites, and then essentially die after a period of time. And luckily, colleagues over at the state diagnostic lab, Woody Frazier, Tom O'Keefe, and Brad Boland figured out, using basically silver-stained supernatant from ultracentrifugation of infected tissues, that it was most likely due to an iridovirus. Again, this is kind of the level of where we were back then. So negative staining, they saw what looked to be an iridovirus, and that was kind of where we were at the time. There was some histo that could be, you could get some information from, but a lot of us were sending them samples just to look for, any evidence of this using that method. Now we know it is actually most of the viral diseases that we're seeing here are megalocytoviral caused. In the Gouramis, we were seeing enlarged spleen, some bloody intestine, amber fluid in the coelom. You can see those enlarged kind of megalocytes here in the upper left, those kind of dark purple enlarged cells. A lot of necrosis in the spleen and other tissues, but depending on the level of infection, these megalocytes could be found in many, many different tissues. And the lower picture is actually from a paraffin embedded block which was run through EM. So I do have to thank and acknowledge Tom Walczyk. When Tom was a student, I remember he contacted me years and years ago because he was really interested in ornamentals. And then when he finished his PhD, I think, around the time between finishing up his PhD. And so, he did spend time with me and Debbie here as a short externship. And he ended up actually helping me a lot while he was at Davis, as well as obviously here in Florida with a lot of the megalocytoviral cases we had. And many of you know him as the herpes guy, but he has more than herpes, let me tell you. So I like to eat, and many times we discuss things over breakfast if these guys are around. So these are some of the infected cases we've had with megalocytovirus, and you can see with the asterisk that we have had quite a few of the more representative, the gouramis, cichlids, live-bearing fish like the platies, swordtails, rainbow sharks relatively recently, as well as I believe a case way back in the 90s, and then even Banggai cardinals, clownfish. Megalocytoviral infections can be found in many number of species, both food, aquarium, marine, freshwater. So it is a big problem kind of in general. All right, this next case is I'm calling green spotted zebras. And this was kind of an interesting case. It's one I really haven't talked about a lot before, but we were called to a farm that was having issues with these kind of green / dark green black spots on the fish. These were a Pseudotrophia species at the time. I think it's Mainlandia now. No other cichlids groups or genera of that type were affected. Primarily larger fish. You can see on the picture some of the dark kind of spots I'm talking about. Really, other than those, and ate normally, no other affected, even when they introduced new broodstock. These fish were being raised in concrete ponds exposed to direct sun. Some of the ponds had a lot of algae, others didn't. That's kind of a hint. This actually ended up being an algal infection of the skin of these fish. And so interestingly, Ruth had in 1994, so these were, I think our case was maybe late 90s, early 2000s. Ruth had a case in 94 of another algal dermatitis, but it was a different, slightly, I think it was the same genera, but a different species, and it was a different algae. So she saw Chlorochytrium, ours was Cenodesmus, and really kind of interesting because I had never seen this really that well before. And even on squash preps and histo, we were seeing algal infections within the skeletal muscle, spleen, and kidney. So even some algae were getting internal, which was very interesting. Because it's an algae, we did attempt kind of algicide type treatments, including copper, diuron, even high doses of NaCl, but nothing really seemed to work very well. And this sort of resolved itself over time, moving some of the fish into different types of grow out areas seemed to help, potentially from aggression or irritation. We assumed maybe some trauma. These were typically the larger fish that would be more aggressive, and potentially secondary invasion is kind of our thought. The picture on the right in the middle is bacterial stain, and you can see that the algal cells did stain positive. I thought this was really interesting as a case many years ago, and then when this article came out in 2019, the path kind of really, I think, covers or encapsulates what I think many of us are thinking about with regard to how to approach diseases. It's really, we do have these primary diseases or primary etiologies or pathogens, but really there's kind of a whole mix of things going on, really, as well as environmentally and biologically within the animal. But I have to say I was actually sort of shocked, but impressed as well that that article was actually referenced in this in this Pathobiome article. So I thought that was pretty cool. And, you know, sometimes you do these weird things and some other people find them interesting, which you wouldn't expect. So that was kind of interesting. All right, discus disease. Many of you may be aware of this because I talked about it many times, many years ago, but not too much recently. This was kind of the early kind of history of this. There were a number of African cichlids, especially, that were being found to have high mortality, these populations. And typically, one of the major findings was granulomas in the stomach, and that was kind of it. We didn't see any organisms, really not much other than that. And that was really kind of a mystery. And I know Ruth had been working on this as well, you know, jointly with when we were looking at a lot of these facilities. In the spring of 1998, this was kind of the turning point for me with regard to this. One facility that was doing discus had these relatively high morts, as you can see, in the younger fish, much lower morts in the slightly older fish, and, you know, really none in the adults. They had just modified their system. They had elevated ammonia and nitrite. pHs were relatively low. These are discus. They were trying to keep them low. You can see the target was 5.8 to 6.2. They were doing water changes to try to keep up with that problem with the nitrogen cycle. And recently, they had added South American cichlids, which was kind of a hint to me that that was probably where the issue came from, but we're not sure. There were some kind of other findings as well, some spironucleus, which is not uncommon. We did see the granulomas, but that was kind of it. And cultures were negative on blood agar. 2 months later, this, you know, and we tried to resolve it maybe by looking at those issues, the spironucleus and water quality, to see if that would help. 2 months later, the mortalities actually increased, and really to a high level in the younger fish. Still above 2 months old, they were very high morbidity. Some of these fish would actually lay on their side, but then after, you know, a week or two, they would be up and, you know, normal again. So they were kind of fighting it, whatever this thing was. The water quality had stabilized. And what we found in this first time, you know, that I had seen was a really heavily infected stomachs in many of these younger fish. So this was suspect, you know, doing kind of lit review and working with our colleagues, especially Sarah Poynton, and who was a protozoologist. We determined it was in fact Cryptobia ubilans, and, you know, that was kind of sort of an alert that this is probably what was going on with the other cichlids, and we just weren't catching it at this really early stage. We did a lot of additional diagnostics and did come out with this paper in 2004. So you can kind of make out on the left side, the histo picture, the little kind of vacuole with a Cryptobia ubilans organism in the middle, and then you can see the granulomas that are essentially, and we had EM done through the granulomas, and they did have Cryptobia within them when they were cut through. We've got some scanning EM as well, shots of these going through the mucosa of the stomach in the middle, and kind of a party of Cryptobia on the far right. So you can see again how inflammatory these are and how destructive they were to the intestinal tract. And these could become systemic as well. We were seeing these in really bad cases within kidney, spleen, you know, just all throughout the body. We did try some clinical treatments. Going down to Miami to get infected populations and running clinical trials was definitely a struggle, because we were trying to get really high prevalences of active infection. I was able to do that with a number of other cichlids, not discus, but quite a few other Africans, where we did verify that it was Cryptobia. And dimetridazole really seemed to work the best of the different drugs we tried. And even recently, we tried a couple of other drugs, and it still seemed to work the best, but still not, you know, necessarily ideal. So this is still kind of an area people can help out with if they, you know, if there's something that maybe completely wipes them out much better, that would be great. So one thing we did find, and we had a grad student that got cichlids imported directly from Africa, put into a sterilized system. The fish had some small and very mild numbers of granulomas when they looked at a subpopulation. Those were held. 6 months later, they were the remaining fish were retested, and they had really heavy, severe gastric granulomas with evidence of Cryptobia as well. So this seems like it may be something that's related to changes in environment, nutrition, et cetera, that will kind of trigger this. We assume, just like spironucleus, these are probably normal inhabitants in some of these fish and will just flare up when there's an issue. All right, so the age of ornamental vaccines. Again, this is kind of the late 90s, early 2000s. I was working with a facility that had really started having major problems with mortalities of the red-tailed black and rainbow sharks. I've got a couple of pictures of the two species there. Primarily post-spawning broodstock. I'll talk a little bit more about breeding. These are actually artificially hormone-induced spawned, as well as in some of the grow-outs in the ponds after they were harvested. Clinical signs included hemorrhages, exophthalmia. Hopefully you can see that pretty clearly on the rainbow shark on the right, and abnormal swimming, as well as lethargy and inaptitude. And yes, this did turn out to be antibiotic therapy is really, really challenging, a lot of limited options. Unfortunately, these are very tricky to feed sometimes anyway, and when they had strep, they really weren't eating at all. This picture on the lower right was one of my attempts to try to figure out how to manage this. We would knock the fish out. I used DMSO with erythromycin, and I sort of calculated trying how to dose them by sort of surface area, what kind of dosing to get the erythromycin into them, because erythromycin and some of the other gram-positive active antibiotics really don't work very well in the water as an immersion, and these guys weren't eating. So this, injection was really not an option with, thousands of fish. So anyway, this was an attempt. It seemed to maybe help, but we really needed another kind of a way to attack this. And that's kind of where we started looking at vaccines. And early 2000s was really when we started looking more toward vaccines, working with Hugh Mitchell, who was, again, a great colleague of mine, and really helped in many ways with us and down here in the industry. So I had a grad student, Ricardo Russo, who was incredible. He did a lot of the work looking at vaccinations. We did primarily injectable. With the broodstock here, we're using one of the juveniles for ease of the study. And he also looked at beta glucans and nucleotides as well. And it was really pretty amazing. After the first vaccination, they really started having very, very low level morts. And after 5 or 6 years of ongoing broodstock vaccination, we really couldn't do anything at the time with the juveniles, other than maybe try to treat without antibiotics, which wasn't very successful. But I think it was more of a reservoir issue, because really after 5 or 6 years, they stopped having any outbreaks at all. And I think a couple of years later, they stopped having to use the vaccine and had no problems for a long time. So this was really kind of our first great success down here with vaccines for ornamentals. So next case is smash mouth. And some of you may have heard of this before, but just to kind of talk a little bit more about it, this was the first time I saw this was in 2002. And it was a husband and wife operation, about a 20-acre facility with one holding building. They were doing other species besides this, which is a tiger barb, but they were having problems with the tiger barb. And hopefully you can kind of see on the lower picture, as well as on the upper pictures, that the mouth really has some severe hemorrhage and ulceration. And this was really the kind of almost pathognomonic sign we were seeing. They would have some spiral swimming and loss of equilibrium as well. These folks in one of the instances did have some elevated problems with their ammonia and nitrite, but it wasn't consistent in all the cases that they had. So I don't, water quality may have contributed to one, but not many of these cases. Bacteriology, and again, this is an ancient photo micrograph or picture of a slide that shows the gram-positive rods, which some of you are probably aware. Gram-positive rods are really not that common in general for aquarium fish. Disease etiologies. This was difficult to sub, grew really fastidiously, at least in our case. And then I had sent it to several labs, including a number of out of state, and no one was able to get an ID. Again, this was like early 2000s, so there really wasn't a lot of good molecular work or that sort of thing done. And so this kind of remained sort of in the shadows for a while, and it would pop up periodically. So fast forward again, breakfast food, again, as you can see, is kind of a theme I like to eat. And I like to talk about diseases. And so this is Esteban on the upper right and Johnny Shelley, who was having additional issues at the facility he was working at the time. And so it kind of reappeared. And we talked a little bit about this. And this was it was now being seen in tetras as well as barbs. Very similar gram-positive rod. We still hadn't really gotten a good ID on it, but you can see in these pictures the extent of the ulceration and damage done to the mouth area of the fish. Wolf, another colleague that really helped a lot with pathology. This is a photomicrograph of a slide he looked at for me. You can see necrotizing facial dermatitis and bacterial colonies, and he saw this And we saw this in other parts of the fish as well, but most severely and obviously on the face or mouth. So when we did get an identity, and Esteban early on, has said it was very close to and maybe Erysipelothryx rhusiopathiae. So that was my first thought was, well, can we use a pig vaccine? I'm sort of getting into this. I had been getting into the vaccine mode, and so for a while, that was kind of in my mind. Would that potentially even work? But as we found out later, no. The answer was no. And Esteban and his grad student Eric Pomeransky did a lot of really elegant work to determine this was another species, different antigens. It would not, rhusiopathiae would not have worked. It was Erysipelothryx piscis. I do want to mention as well that Justin Stillwell, who now I think who is actually at Mississippi State now with Natalie. Congrats to them for their new position. Also detected it in Gambusia in Mississippi as well, Erysipelothryx piscis. So this is kind of an emerging disease and something to keep an eye out. I did want to point out as well that Erysipelothryx rhusiopathiae, Esteban showed can be a pathogen in fish, although at least in the ornamentals, it was more likely to be a piscis for species. Over the years, again, talking about vaccines in general, we've done a lot of work with Hugh and some with Esteban, a lot with Hugh, looking at developing vaccines, autogenous vaccines for various outbreaks we've had. So done definitely a lot of work with Strep A and some other streps, Vibrios, emergent type vaccines, and oral vaccines with Eek delori, and as well as some others. But these were, I think, kind of really, really important for some of the issues that we were having, and hopefully we'll continue to develop even better vaccines as well. All right, so a little more current, Tetra's tiny problem. So this was another facility that was having issues in October 2017, the first time we saw it. Chronic morts of these black tetras, especially after a major stress, so after harvest and during holding. No obvious external parasites or even obvious parasites on wet mounts internally. We did see very small dark cysts in the intestine. Cultures were negative on blood agar. So of course we did submit for histopathology and save tissues for virology as well. On histopath, and I'm not a great histopathologist. I did see what I'm familiar with what DIT dots look like, and they look like DIT dots. You can see in the upper left picture, these little tiny punctate kind of eosinophilic, bodies. And so, I wasn't quite sure. Jeff Wolf suggested lunistain, and you can see on the right, actually stained the microsporidia. So this was a really, really different and weird presentation of microsporidia. The upper two pictures are from the liver. The lower pictures are from the testes of an infected tetra. And you can see that they are, fairly spread out. They're not kind of clumped in as we see in some of the other species. The microsporidia were fairly spread out throughout multiple tissues, as you can see in the lower bullet there. So in the eye, brain, gills, stomach, liver, etc. So kind of all over the place. So I definitely want to thank Jan, and I think Jan might be on this, might be watching as well. So thanks, Jan, for all the help as always for everything. He has done a lot of work with other organisms, but definitely with Bacteria. So he kind of helped remind me, these are obligate spore-forming unicellular. They're either fungi or sister to fungi. A lot of described species that infect a whole host of taxa. And they don't have mitochondria. They have a mitosome, which is, does not have DNA, and they're still sort of figuring out what it does. And they will evert their polar tubes and filament to inject the infective sporoplasm into the host cell. And so on wet mounts, on the left side, you can see this kind of glugia light, I'm calling it until we get to the next slide, but you can see how the microsporidia are sort of spread out a little bit versus the middle picture, Plastophora, which is from one of our cases with swordtails, and then a fathead minnow picture that Ed Noga let me borrow from his book with glugia, and you can see again the xenoma formation in the body cavity. So definitely a different presentation of microsporidia from what we're used to. And with a lot of work and a lot of collaborators listed here in the paper, we had this come out officially this year. It's tetradisseminated microsporidiosis. Jan led us all, and although it was closely related to Glugia as per work by Tom Walczyk, Jan is proposing or has proposed it as a new species, Fusosporus stethoprione. Fusa means spread out, and Stethoprioninae is the caracid family that was a host in both our case as well as in cases that Justin Stilwell and Al Camus saw through the University of Georgia in the cardinal tetras. So really, really interesting. But, again, part of my job is to figure out now what do we do, So knowing what the problem is really complicated question. These were not broodstock in some cases that could be just depopulated for various reasons. So we're obviously talking about biosecurity and trying to get a clean lineages for these groups as well down the road. But immediately looking at disinfection and sanitation, and we attempted a couple really rough trials, pilot studies with chemotherapeutants. There are some in the literature for microsporidia, including the benzimidazoles, fumagillin, which was no longer available. I'm hearing that it may be available again. I'm not sure. It was used for microsporidia in bees. We ended up getting fairly good success, at least in terms of survival of the fish with toltrazuril. We haven't really done the next step, which would be looking at maybe EM and other level cuts of the fish after they've been treated to see if it's actually infecting or affecting the microsporidia. But we have had, again, success with toltrazuril so far. All right, so talk a little bit about drugs now, and this is kind of from Matt, because as many of you know, Matt really likes drugs. So we have one of the big things we've worked on here at the lab, both me and Debbie and folks in aquatic animal health, as well as Craig and everyone here in general on the production side, is really trying to develop and legalize drugs for the aquaculture industry. And as some of you may know, it's very, very challenging to get drugs legalized through the FDA for various reasons. So that's always been one of the big reasons that is sort of not a therapeutic, but more of a production reason for drugs is induced spawning. Back in the late 80s, Dr. Shireman, who was the chair of the department that fisheries at UF, and Jim Gilday, who was his grad student, figure out a way to induce spawn using artificial hormones, rainbow sharks and red-tailed black sharks. And this was a huge breakthrough. Now, this had been done in salmonids and other species, before, but this was really the first time it had been done in ornamental fish. And the reason they were using it was because they could not naturally or environmentally spawn the rainbow and the red-tailed black sharks, which were really a popular hobby fish. So this was a huge thing. And they were using different chemicals, et cetera. They were really not established as drugs. And so I just wanted to talk a little bit, real briefly, again, on artificial hormone induced spawning. It has opened up production species. I mentioned the red-tailed black shark on the upper left. We've got tinfoil barbs in the middle, iridescent sharks below that. On the right, we have the clown loaches. Craig and Amy here at the lab were the first to do both the clown loaches as well as the spotted green puffer in the middle. And then we've got farms that are doing a lot of the Synodontis catfish, which are a really cool group of African catfish on the lower right. In the middle, a lot of these fish are going to be similar to, I guess, some of the other species. And on the food fish side, after they're spawned, they're going to be stripped and put into these modified McDonald hatching jars where the larvae and the eggs are going to kind of hatch out and develop until they're ready to be moved to the next level. So one of I think the big successes we had here was really working with both Sindel and Western Chemical at the time to look at overprim, look at its use, and it was really being used fairly widely and had been for a long time when it became available. It was legal in Canada, not legal in the US. So we worked really closely, and after FDA was able to get the Minor Use Minor Species Animal Health Act of 2004 passed, that allowed for the indexing method for getting legal use of drugs. So we went ahead and I was the chair of the expert panel for that, and we were the first. Overprim was the first drug to be put on the index for FDA, and so again, that was a huge kind of relief of burden. There was a lot of paperwork involved prior to that. And so, we did a lot of extension outreach for the producers after it became available through legally through Sindel. So you can see it's indexed legally marketed unapproved, so they can use it legally, but it's as per the index, it's not approved, but it's still legal. So very confusing, but it was a good thing. We also did a lot of work with Metomede, which many of you know is a sedative used in transport and for some other purposes, and shipping. So we did a number of studies for this, and we're actually able to also use it for just recently, looking at it as a use for euthanasia. So Aquacom as a sedative was the second index product of any species on the label as well. So we were proud of that. Craig Watson was, I think either the chair, I think it was the chair for Aquacom, or Jim was on that as well as another colleague. We've also did other work. I just threw one up randomly. We did an oral study with Slice, emamectin benzoate, which really worked really well against Argulis when we were trying to work with it was sharing Plow, Intervet to look at maybe getting an index for that. It hasn't gone anywhere, and this was a while ago, unfortunately. Again, antibiotics, as we talk about many times, unfortunately, antibiotics, a lot of the doses we're using are either based on empirical information or on food fish studies. And so Greg Lubard, a number of other folks have really looked at trying to up the number of studies looking specifically at ornamentals. And so I did some work with chlorphenicol in koi carp in gourami back in 2005, actually before 2005, but the paper came out in 2005. And then recently with Brian Borbach, who was one of our interns and was at South Carolina Aquarium for a while, we've done a number of studies with giant danio as a proxy for zebra danios and some other smaller bodied fish. This paper is on Oxytet. We recently did chlorphenicol and rofloxacin as well, using kind of this sort of microhematocrit tube blood letting method. So kind of takes us to the end here. So some deep blue thoughts I had. Diseases are like fashion. They always seem to come back. And that's also for, that was also for Matt. So technological, biological, bioinformatics, and digital advances have really pushed our understanding and our approaches to disease and health management in aquaculture. I really owe huge debt of thanks to very, very smart mentors and friends who have helped in many, many cases, even if it's just working a specific case, not necessarily a paper, but in collaboration. I do want to point out aquaculture and agriculture are really dealing with more than one type of climate change. In addition to the outside climate change, there are definitely issues with science versus social and political misinformation. And I think we kind of need to up our game PR wise in many, many ways, unfortunately, to try to help take care of that. And finally, variety and complexity in the industry will continue to make a lot of the health and disease work very challenging and exciting and in need of others. And some of the things that we are still looking at include just general efficiencies in production. A lot of things I could go into about that. Really a lot of information with all the species we're working on, looking at specific biology, physiology, nutrition. A lot of work, Tom and Esteban and others looking at disease balance in general. We are kind of trying to look at diseases of broodstock and grow out, especially some of the marine larvae. It's really a challenge. Biosecurity is a huge thing. Vaccines, of course, others, the people are interested in bacteriophages again to a degree. Probiotics, we were working on a USDA grant with Matt Griffin and John Hawk and others, kind of looking at a multi-pronged approach to E. coli, and our end is looking at some probiotics with Chris Martinick, who's up in Gainesville. So I think interests in probiotics are continuing. More drugs, of course, want to continue to increase our knowledge of the different drugs as they apply to our species and not food fish. And then, of course, logistics and compliance and really looking at the human side of things, like how can we help to come up with ideas that the farmers are going to be able to do logistically as well as, understand. USDA has the CAPS program, Commercial Aquaculture Program Standards, which they're trying to and have been sort of trying to work out to help kind of bring everybody together as a team for aquaculture. Again, thanks to all of my smart friends and colleagues, a lot of different groups that I belong to. For the students, I know many of you are, I'm sure, familiar with many of these groups, but really, I've made really, really great friendships and have gotten some great insights and help over the years from all of these groups. And I definitely do want to thank Debbie Pooter, who manages the lab here, and Eric Curtis, who used to manage the lab here, as well as all past and current faculty and staff at our lab. And with that, I think I appreciate everyone listening. This is Halo Hala. If you're not familiar with it, look it up or try it if you're ever near a Philippine. Tasty, it is brain food. And I think there may be time for some questions.
There sure are, Roy. Thank you very much for the awesome talk, interesting and entertaining as always. Folks, feel free to unmute yourself if you want to ask Roy a question, or you can drop them in the chat box too. All right, you get some compliments in the chat there, Roy. I can kick things off here. I'm curious to know a bit more about the maybe the proactive health management that you do. You talked a lot about a disease case presents itself and you have to figure it out, but what sorts of things do you do, especially for that tropical fish industry where you have very little baseline and fish are being imported and moved around a lot?
Yeah, it's definitely complicated. One of the big challenges, I think, is understanding the facility and how they how they do management. We did get a grant, kind of looking at biosecurity and trying to sort of come up with sort of a program. As you can imagine, it can be kind of complicated to try to, biosecurity, I think, is kind of like insurance and kind of actually like using plastic covers for a lot of farms. They're going to spend a lot on these plastic covers, but they're not really going to help much unless there's a freeze. And when there's a freeze, all the fish die. So it's really challenging. We do have farms where we'll talk about different aspects of biosecurity and different ways that maybe they can help prevent disease. And we do have some takers, but it is kind of as probably in general, like until like there's a calamity, it's really hard for sometimes for major changes to occur. But we do have we try to push the ball inch by inch in some places and even further in others. So I think a lot of it, we do a lot of outreach programs. We have a 2-day fish health management class for targeting the producers. We do site visits and obviously get a lot of phone calls and people, emailing and that sort of thing. So I think the biggest thing is just trying to increase their awareness and talk about, how everything is interrelated. And, that's kind of where we are with it. We are trying to work on other ways, trying to work with caps to a degree, on some facilities if we can, but we're still kind of at sort of a gray area with regard to, where the industry is versus, like your average shrimp facility, or some of these other much higher level type biosecurity.
Awesome. Okay, Tom's got a question.
Yeah, Roy, awesome talk as always. So for me, I know we've got a lot of graduate students and people in various stages of their careers attending. And to me, like, your talk was awesome at demonstrating how important collaborations and all the networks of folks that you've worked with. I mean, that rang true. I wonder if you have any just advice on that note for some of the newer people coming up on how you're able to make that work so well for you.
Yeah, definitely. And that's a great question. And yeah, Tom knows and Matt knows and a lot of you guys know. I'm not as smart as many of you, which is fine, and I accept that. I think a lot of it is really kind of knowing when you need to seek help, and really getting involved with a lot of the organizations, whether it's AFS Fish Health or Eastern Fish Health Workshop or some of the other groups that you may be, to really start getting your feel for where people are working and what areas there are. That was, I think, one big thing for me. And again, as Nick mentioned, I'm one of the main organizers for the Eastern Fish Health Workshop. I really kind of learned a lot of where people sort of spent a lot of their time and what their interests were, and it became really easy, and became really great friends with a lot of these folks, just to call them and say, hey, I've got this problem, and what do you think, or can you help? And a lot of times the interest is there already, and they're more than willing to even maybe make a project out of it to learn more about it. So I think a lot of it really does go to going to the meetings, figure out what other people are doing. I think it can be easy sometimes to be in sort of a silo or one area. And, a lot of these meetings will expose you to a lot of other areas, and I think that's kind of one thing I really learned was, just because I'm doing what I'm doing doesn't mean that somebody, maybe doing, something completely different won't have anything to add. I kind of see everything as related. So I think maybe make sure you're not kind of boxing yourself into something. And I think that goes with research as well as diagnostics, as well as, education in general. So really, yeah, try to go to a lot of meetings, meet a lot of people, and don't think that because they're not doing something related to you that it may not actually be interesting. They may not be, that they wouldn't be an interesting person anyway, but you may learn a lot from just their way of thinking. And that's kind of how I approach all of this.
Good advice. Thanks, Roy.
Thanks.
Hey, Roy, this is Mark, as always.
Thanks, Larry.
Just kind of leading into what Tom asked about, are there that you find that information that you can really use in your work in Florida?
That's a great question. I think I've tried looking, but I may not have been looking as hard. I think our the way we work extension is so different that I think there, I may be potentially not approaching some of these other things in the right way. But I haven't really seen too much, maybe working with like Tom or some of the other folks that are working with folks internationally. I've gotten maybe some feedback and ideas, but that's a great question. And I know, I've spoken to folks in other parts of the country, but I think a lot of times there may not be some of the things that they're working on may not be quite. Again, that's definitely something that would be of interest, if folks are working on other things. Like, for example, in Singapore, I really don't know people. And I know they're doing a lot of work in Singapore, but we just don't really communicate I think some of it may be just related to impressions and in terms of like competition, that sort of thing. So it can be a challenge as well because of the way the industry is structured.
Hey, one more question. How have the changes in the tropical fish farm industries in Florida, how are the changes over the past 20, 30 years? How's that changed how you do your work, or how you think that those of us in fish would be working with ornamentals in the future will be doing things differently than maybe you did when you first started?
Yeah, that's a great question. I mean, just in general, I think the industry is really consolidated because of, the mom and pop stores sort of going away, and PetSmart, Petco, Pet Supermarket, Pet Supplies Plus, all these other kind of larger chains taking over. The species have changed a bit. I think the advantage is there's a lot more people interested in working with fish, and there's a lot more knowledge than there was back, when Myron, when you and I started, definitely. So I think there are a lot more tools and a lot more laboratories working on areas, whereas, back 10, 20 years ago, there really weren't. So I think the opportunities for, again, part of it is to Tom's point, networking with folks that are working on specific areas that may be outside of your purview, but also, being exposed for a lot of the new folks to these meetings and kind of knowing what is out there right now. There's a lot more kind of references for fish disease in general, definitely, as over the past 10 years and 15 years than there were for us. So there's a lot more documentation of diseases as well. So I guess that's not really a great answer, Myron, unfortunately, but I think there've been changes with regard to knowledge base approaches, personnel and diagnostic labs, as well as and I will say, as well as the approach in terms of the, on the hobby side, we get a lot more calls from hobbyists that really want to work with a fish health person. We have a lot more, I think, understanding just by the fact, that fish are animals, that, that there may be ways that we can work on them or work with them, versus being more kind of a disposable sort of commodity. So I think the attitudes have changed a bit as well. Although, you know, obviously it will kind of vary from person to person. So I think there's kind of been a change both industry side, you know, socially, as well as with infrastructure for health management in general.
Roy, Dave Scarf here.
Dave,
Nice presentation.
thanks.
Exercise your great wisdom here and tell all the young folks that are wanting to get into ornamental fish medicine, what would you recommend for them today?
That's a great question. Some of the things, and interesting, you know, we do have a lot of externs that come through the lab that are interested in doing, you know, hobbyist type work or aquarium fish. And I would say, obviously, be familiar with sort of the basics, but you know, kind of develop sort of an expertise with some of the we call domestic fish, you know, like koi and goldfish, and have it, you know, set up an aquarium. I mean, that's kind of a sort of maybe obvious thing, but I know some folks that really haven't set up their own aquarium to just sort of get a little more familiar with that. Also, we talk about trying to let yourself be known out there. There are actually there's some publications, and I can talk to whoever is interested on like what sorts of things you need to sort of set yourself up to work with a hobbyist in terms of equipment and supplies. But yeah, I think, you know, let yourself be known to local pet stores as well as aquarium stores and aquarium societies. You know, that's kind of one thing that I think will be very helpful. And of course, you know, attend meetings and keep networking with regard to increasing your knowledge base, continuing ed programs, et cetera. Really important, I think. And, you know, if you can shadow people that, you know, don't mind or, you know, work at volunteer at a public aquarium, you know, if that's possible, you know, or even work, you know, with a pet store, et cetera, that may be another way to kind of increase your knowledge.
Great advice, Roy. Thank you very much again. We're at time, so we're going